So the concept of infection is our second concept, and the hidden concept, of course, is going to be immunity. So infection is going to have inflammation, is going to have the immunity concept also playing a big role. Um, Wherever we go, we find organisms all around us, um, some of them are beneficial, some of them are harmful. We share basic functions with those, and that's survival and reproduction. Um, like I said, there are some organisms that we host on our skin and our mucous membranes, and there's called normal microflora, and there are others that are just um, pests that we don't want, and these if we don't have the immune system that's able to fight and eradicate them, they can be harmful and potentially lethal. So in the study of the infections, we usually look at when we're looking at infectious diseases and infections, we look at it. We There are variables that play in it, and three main variables: the agent that is causing the infection, what type of microbe it is, whether it's a virus, whether it's a bacteria, whether it's a fungus, and then the host and the environment that it's gonna survive in it. So we'll talk about these as we go in this concept. First, let us define the concept. This is the definition of the concept of infection from the Giddens book. And when we talk about this concept, we really want to present the risk factors, what signs and symptoms you can see in an infection, and how do you treat and manage the infection, but also the infection control and prevention aspect that we covered early on in the semester. So we're going to talk about these key terms in the lecture. What will these different terms mean? And as you have heard me state in the beginning that there are three main factors that play in the infectious disease process: the host, the environment, and the agent. And these are covered in other disciplines other than the medical field. Epidemiology, microbiology, immunology will talk about these same key terms. So we'll talk more in the class. Um, how do we categorize infections? So um, we said that we have the viruses, and viruses can be both DNA or RNA, made of DNA or RNA. We have the bacteria that's mostly DNA. We have mycoplasms that is mostly DNA. There are the spirochetes, the rickettsias, the chlamydias, the yeast, the molds, the protozoas, the helminths, and also there are the prions, which are thought to be of some bacterial origin, but they're like um, not defined really. Uh, they don't have a genome to them that can help fight, and that's why they're very resistant, and they present a significant a challenge when it comes to infection control. When we talk about the scope of infection, again, we need to speak anywhere from free of infection to infected. And then that also we talk about additional terms in here in the concept of infection. That is the epidemic and the pandemic. And so what is epidemic is it's where there are some infectious disease that are normal for the population or the geographic area, but there are just more cases of them at this moment. A pandemic is when there is an epidemic across the globe. It's a worldwide, if it's more of a global nature. So that's what we are experiencing now, and you'll be the cohort that understands it because you're living it more than other people. Um, There are several descriptors of infections. One time we talk about an infection being epidemic versus pandemic, and we'll dwell more on that in the classroom. Versus when we talk about where in the body this infection is, what body system is affecting, versus what mode of transmission it is um, uh, transmitted through, versus the illness, like is it a deadly infection? And then we talk about bacterial infection, where bacteria are those one-celled organisms that don't have a nucleus really, versus the viral infections that are caused by that virus pathogen that has the nucleic acid and that needs that host for replication. And then you know about those fungal infections where you see yeast, molds, mushrooms, protozoas, parasitic infections. These are usually generally do not infect the general population. You have to have compromised immune response for them to really be virulent. And sometimes the infection will start in one place, and then you'll get a secondary infection after that. So you have a bacterial infection, they give you antibiotics, and then you develop fungal infections secondary to that. That's when they talk about it. And sometimes you come to the hospital with no infection, and then the healthcare system gives you an infection. That's where they'll call that healthcare acquired. Versus if you came to the hospital with it, they'll say that's a community acquired. And then they'll talk about those resistant infections. So methicillin resistant Staphylococcus aureus, which is the MRSA, or vancomycin resistant enterococci, which is the VRE, and the Clostridium difficile, which is the C. diff that you know. And these have been placing a big burden on the uh, healthcare system. And lastly, I want to highlight with the location of the infection. Most often, the infection starts in a place and then disseminates throughout the system. Very rarely, you will find it directly in the blood and affecting all the organs, unless if we really have a central line and that infection goes directly in the blood, or with IVDA users and that goes into the blood. But otherwise, you start with the UTI in the urinary tract system, and then it goes to the whole urinary system. You call it urosepsis, and then goes to the whole body, and they call it sepsis. So we'll talk a little bit about endemic, epidemic, and pandemic. We already covered the immune system, and I would like, if you want to go and watch that video. I posted my video. I posted a small snippet video about immune system, and then do read your book, because the concept is not covered in this uh, course, but it is a backbone for these two concepts that we're talking about this week: inflammation and infection. So the process of infection is represented in your book with the figure 24.2, and it shows the interactions of the three main elements: the host, the environment, and the pathogen or the agent. And you have those interacting together, where you'll see six elements, which are the pathogen and the susceptible host and the reservoir. And then you need a portal of exit from that reservoir, and then you need a portal of entry to the susceptible host, and you need that mode of transmission. That's why we talk about infection control. When we talk about precautions, we have the standard precautions, and then we have the contact precaution, the airborne precaution, and the droplet precautions. So if we look at these three elements really in a little bit of critical thinking, we see that the infectious disease will only result if the agent or pathogen have those favorable environmental conditions that promote infection within that susceptible host. So if the host sup- why we're called host, because we are providing the nutrition and physical growth for that organism or for that agent. And there's a concept I want to introduce to you here is the concept of colonization versus the concept of infection. And these are used interchangeably, but they should not, because infection is the presence on and multiplication within a host of another organism that causes injury to the host, whereas colonization is the act of just that agent is just present, and it needs to multiply in order to say that there is an infection. And again, not all interactions between microorganisms and humans are detrimental, because you remember we need to know that our normal flora in the GI and on the skin are actually protective. So when we talk about pathogen invasion, we're talking about how the infection is happening, and I need you to be aware of that, because if you don't know how the infection the pathogen can invade, you're not going to be knowing of how to prevent that invasion. So you need to know the modes of transmission. First mode of transmission is penetration, and penetration is when you disrupt the barrier that we have, which is the skin or mucous membrane. That's a natural immune barrier. And so anyway, anytime we do that and we do an invasive procedure, such as putting a peripheral IV or a central catheter in, we are giving the potential, a potential portal of entry for the microorganism to come and invade the host. So that is one way. The second is direct contact, where, for example, sexually transmitted diseases are directly transmitted from one infected tissue to another infected tissue, although you may have also intact mucous membranes. And so gonorrhea, syphilis, chlamydia, herpes, they're all um, uh, uh, um, transmitted via intimate contact. The other way of transmitting is, for example, ingestion. When we eat something via oral, we'll get, for example, food poisoning, or cholera, where they get it from contaminated water, or hepatitis A. So these are caused when you have when you ingest contaminated food or water. And another method of transmission is inhalation, and that's through the respiratory tract. We have a defense system along the respiratory tract, and those are those cilia, the cough reflex, and all those. But sometimes there are pathogens that are able to invade the respiratory tract and cause bacterial pneumonia, such as strep pneumonia. You get also Haemophilus influenza, and you may also get a chronic infection such as tuberculosis, as well as other viruses. You remember the MMR, the chicken pox, also they're all in that list of um, um, inhalation route. So when a pathogen, be it a bacteria or a virus, or whichever pathogen invades, the body will initiate the immune response, and both types, or all types of the white blood cells, are gonna get into the equation to try to fight, from the B lymphocytes in producing antibodies to the lymphocytes that will orchestrate all those T lymphocytes peptides that orchestrate all the fight against an infection, and the complement system will be activated, and you'll see the inflammatory response secondary to those toxins that the bacteria or the virus or all those will produce when they invade. The body sees them as antigens, and then it will activate the immune response the um, um, in the regular response that it has been built to do. What happens is if there is uncontrolled infection response, so the infection either gets untreated or it doesn't respond to the treatment, and the inflammatory response goes haywire, you're going to have the body that can't go back and restore itself, and the compensatory mechanism that the body goes through from the cardiac to the respiratory to the nervous system to the kidneys will be exhausted and will be consumed. So you'll see those multi-system failure because the invasion of the microorganisms invades the whole body. Typically, it's with an bacteria infection, but it can be with anything. So that the patient will end up with multi-organ system failure, or you'll hear it with MODS or septic shock. And that is first they'll get into sepsis where the infection is throughout the body, then they go into shock where they have ineffective tissue perfusion, and eventually first the septic shock happens with one organ being involved, and then when multiple organs are involved, the chances of survival dims, and then death is will occur when the body cannot recuperate from that injury. Risk factors are really similar to the risk factors in the inflammation. You have those certain things that put a person at higher risk. That's a very young and probably the very old, but poor, uninsured, certain geographic areas, and that comes from the infrastructure as well. So if you have poor water supply, that brings along cholera and so many things. Like malaria is present in certain places in Africa because of the presence of the mosquito that carries it. So that when we talk about the vector, so there are things you can't change because of the geographic location, but mostly a compromised host with either an immunodeficiency or a chronic illness, those with diabetes, those with um, uh, heart failure, kidney failure will be your patients who are at greatest risk for getting infections. So we'll talk about bring some of the list of individual risk factors and some environmental factors that you know about when we're in class. Also, we're gonna talk about some of those symptoms and clinical finding that we see in specifically in wound infection. But I'm going to talk about the general clinical manifestation. So this is the signs and symptoms that the host is going to show during the infectious disease process. And those can be anywhere from diarrhea to rash, to fever, to headache, to lethargy. And so some of those symptoms can be um, non-specific, but there are symptoms that are specific. You remember with chicken pox, you're gonna see those skin lesions, and or in measles, you're gonna see symptoms, but mostly there are some covert symptoms that you only can get when you do lab testing. So we'll talk about some of those lab testing. Okay, when we do CBC, we're looking at the white blood cells. So we're looking at those exact white blood cells. How is every one of those white blood cells acting? What's their presentation? You need to go back to your anatomy physiology and to, if you want to listen to that lecture, to hear more about what does the serology test of getting the CBC mean, and why do we do the differential? And then you get the cultures and sensitivity. What do they mean by culture is the growth of microorganism. So they'll put them on in agar plates, and then they'll inoculate the uh, whatever they took from the host, and they'll see what kind of organism grow. And the gram stain, if they're positive, that means it's a positive gram-positive bacteria. If it's negative, and or the different types of bacteria. The other culture test they do is the CRP. And CRP, ESR are inflammatory markers. And then you have those serology tests, or what they do, they sometimes do the antibody titer to check for exact specific antibodies or specific viruses. The PCR, for example, in the case of the COVID, is been tested as been used as a serological test. Radiographic testing, whether X-rays, MRIs, CT, or a PET scan, they're usually not confirmative in and by themselves, but they're used as supporting treatment, so that can help identify the area of the infection or the area of the inflammation. What is the best management? Like we spoke in the inflammation, we can always have primary prevention, prevent this from happening. And for that, we talk about vaccination. You get the gist of immunization in your 115 class in your first farm, but you need to be familiar with what vaccination is. And if you listen to the lecture in immunity, you'll learn that vaccination is an active acquired infection. I mean immunity. I'm sorry. So it's an acquired but active immunity, because they give us the attenuated organism, and we as an antigen, and we build the antibodies against them. That is one of the primary preventions. Another prevention is hygiene, personal hygiene, food hygiene. So when we talk about personal hygiene, we're talking about the general personal hygiene and hand washing. But when we talk about food hygiene, food handling, food storage, and all those things that will really help prevent gastrointestinal infections. And we covered this infection control practices initially, so go back and revisit that chapter. Uh, we're gonna discuss a little bit hand hygiene and see if we have any questions on that. Secondary prevention. Can we do secondary prevention in this case? So here we can do so many things, because really we can see who's infected and we can prevent other people from getting infected. For example, that's one thing. The other one thing is, for example, people with the AIDS disease, they're at high risk for all opportunistic infections. So, to prevent, like to help them to not get in contact with somebody with that infection. Or people who are in high risk groups, you teach them how they use safe sex, so to prevent sexually transmitted infections. So when we meet, I want you to tell me who's a high risk. Okay, then there are those all nice treatment, and the treatment here is the removal of the pathogen from the host and restoration of that normal physiologic function. So when you treat, you have to treat really on multiple layers. So first and foremost, antimicrobial agents. When they say antimicrobial agents, antimicrobial therapy, they're talking about therapy against more than one organism. So you talk about antibacterial agents, and then antiviral, and so on and so forth. In antibacterial, for example, there is that bactericidal versus bacteriostatic. For example, bactericidal is an antibiotic that is lethal to that bacteria, versus bacteriostatic is just an agent that will prevent the growth of the bacteria. But if you remove the agent, or the therapy, the bacteria can regrow. So that's static, just put it in a static mode. And you'll learn about my antimicrobial therapies. They usually are, there's multiple families: the penicillins, the cephalosporins, the carbapenems, the aminoglycosides, tetracyclines, macrolides, quinolones, and so on. So there's so many families that act on different levels of the organism. Some will kill their cell wall, some they will um, prevent protein synthesis, some will work on their ribosomes. So the different methods of um, kill factor for the microbe. And then rest and comfort measures. So when we talk about rest and comfort, the body is fighting a very strong um, enemy, and then it's gonna use all its protein resource. Fever is gonna dehydrate the patient, and so you need to give them time to rest. And but you'll work on the nutritional support. So a diet that is rich in proteins, and as well as a diet that is replacing those electrolytes, because of the fever, the vomiting, the diarrhea probably that's happening, and fluids replacement is essential. Then the disinfection of the physical environment. So we talked about this a little bit uh, previously, but really we shed ourselves. So now this host is shedding, and they're shedding in their environment. So you need to clean the environment with the appropriate disinfectant. Again, interrelated concept. We highlighted immunity as being a critical factor, and inflammation, because when we have infection, there's going to be inflammation. Tissue integrity, stress and coping, nutrition. These are concepts that are going to be intertwined with infection. What are some of the exemplars of infection? We need to discuss those, and I would like you to look at your book. Your book talks about pneumonia, conjunctivitis, otitis media, hepatitis, just in simple knowing what does that, what all those infections mean. Okay, so and human papilloma virus is that's the HPV, the sexually transmitted infection, and then they're fighting it so hard because it's been found that people who contract that virus, they have a higher rate of cancer. So that's why we have that HPV vaccine that is encouraged for people between the age of 9 to 14 to get it.